So we're going to go over how to read a scientific paper, specifically one of these papers dealing with pigments in the fruit fly eye. So we're looking for genes responsible for building molecules or transporting molecules related to eye pigments. And each paper typically focuses on one gene, and we'll be looking at mutants for the gene and getting at the function for the gene. So when we talk about gene function, the gene is the DNA, and the DNA from that gene is the instructions for making a protein. And that protein, in the case of eye pigments, is going to be a transport protein or a synthesis protein. And synthesis proteins are also a form of enzymes. And so you might see discussions about enzymes, as in this paper. You also might see descriptions of locus, as this is written here. So this is a paper we're going to talk about dealing with a gene called Rosy. Locus is another way to refer to gene. So locus, meaning place or position along the chromosome, means that there's a certain piece of the chromosome, piece of DNA that contains the instructions for making the protein that results from Rosy, the Rosy gene. So when people were discovering genes and discovering gene functions, they might start with something that's known and a whole lot that's unknown. And typically early in the process, they would have a mutant phenotype. So that's where a lot of these genes got their names. The Rosy gene comes from a specific eye color that results from the Rosy mutation or mutants of the Rosy gene. And sometimes they're different from other ones because they need to be unique. So this might be a color that's similar to another color, say purple, but it needed its own name. So it gets called Rosy. As we've seen, not all the different names for the enzymes, the different names for the genes are really all that different in color. All right, so getting into this paper here, I might have searched to find this paper using the name of the gene, Rosy, the name of the organism, Drosophila or Drosophila melanogaster, and then maybe another keyword like function or enzyme or something like that. So most importantly, really, when you're finding papers, would be the title and the abstract. And these are going to tell you in the quickest amount of time whether this is a paper that's relating to what you want to learn. So this title is pretty informative, tells us we're talking about the Rosy gene, tells us we're talking about the model fruit fly, Drosophila melanogaster. And then that last bit at the end mentions eye pigments and also mentions xanthine dehydrogenase. And you might notice the standard suffix for an enzyme. So xanthine dehydrogenase, we don't really know what that does right now, but it might be related to this Rosy gene. So title, authors, of course, publication year, and then abstract. So the abstract is basically a little capsule encapsulation of the paper. Should include components that you normally find in the introduction, parts that you normally find in the methods, parts that you find in the results, and parts you find in this discussion, all sort of summarized and really condensed down so that a person can understand what this paper is about, really decide whether it's worth reading for their interest without investing a whole lot of time finding out about it. So let's go through the abstract here. The Rosy gene in Drosophila melanogaster codes for the enzyme xanthine dehydrogenase. All right, so if you didn't know that already, that's pretty helpful. So we have a name for the enzyme that results from the Rosy gene. Enzymes often get names that are informative about their functions, so it probably does something related to xanthine dehydrogenase. Mutants that have no enzyme activity, so these would be recessive mutants for the Rosy gene, are characterized by a brownish eye-colored phenotype. So that would be the so-called Rosy phenotype, reflecting a deficiency in the red eye pigment. So as we'll learn, there are two major categories of pigments, the reds and the browns in the fruit flies. And so this is one that's deficient in the red category, and therefore it shows more of the brown tint to the adult eye. All right, xanthine dehydrogenase is not synthesized in the eye, not made there, but rather is transported there. And this present report, as it says, describes the ultrastructural localization of xanthine dehydrogenase. So basically this study, as we get in 3-4 lines in, tells us that they're interested in knowing where the enzyme is found in the developing fruit fly eye. So actually, prior to this study, it was pretty well known, it looks like, what the enzyme did that was made by this Rosy gene. And then this specific study gives us some primary firsthand evidence for where the enzyme is located and how that relates to other mutants for the eye pigments. Okay, so let's go ahead and jump in. I think we've probably gotten some pieces of what's going to be in the paper. And we'll get into the introduction. Sometimes the introduction is labeled. Sometimes, as in this paper, you just jump right into the text. So the first text after the abstract would be the introduction. And of course, the abstract is set apart as its own small paragraph. Again, not every paper calls it the abstract. They might just have this little paragraph here. That's what it is, and that's pretty standard. Okay, so introduction. The major parts that are included in the introduction are background information, and then kind of leading up to the motivation for the current study and possibly some hypotheses for the current study. So as far as summarizing what is known, so I guess they start in what is unknown. Despite many investigations, yada, 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 the real understanding of the production of eye color pigments remains elusive. Dun, dun, dun. The Rosy gene of Drosophila melanogaster is one of more than 83 different genes for which mutations are associated with an altered eye color. So you and your classmates are looking at lots of different genes related to building up the eye pigment molecules. Rosy codes for the enzyme Xanthine Dehydrogenase. So right off the bat, this is something that was already known before this paper was published. And this, admittedly, for the level that we're interested in, this little sentence here might be one of the most informative take-home messages from this paper. It's been the subject of extensive study. Homozygous null mutant adults. Null means a completely non-functional or even absent allele for the gene. So homozygous null mutant adults, or homozygous recessive, you could think. Rosy adults are characterized by a brownish eye color. And now it's a little more specific here, a partial deficiency of the red pteridine pigments compared to relatively normal levels of brown xanthomidin pigments. So these are names you might also see in other papers, pteridine and xanthomidin. And those are the names for categories of pigment molecules. So molecules that show red color or molecules that show a brown color. All right, so XTase catalyzes at least one pteridine reaction. It has never been shown to catalyze a reaction that in the pigment pathway that takes place in the eye. Okay, so the kind of interested in this study to know where the enzyme is acting and maybe how it gets there. So I guess I don't want to go into great detail here. This is one that we normally in person would cover kind of piece apart and cover the paragraphs individually. But I think I want to go a little bit less detail this time around. So still in the introduction, lots of background. Here we're getting a little more specific. Next paragraph, in a prior report, we demonstrated that the Rosy gene is transcribed and the enzyme is synthesized in fat body, malpighians, tubules, and gut cells of larvae in adults. But the Rosy gene is not expressed in the transcript level in cells of the adult eye. So it's kind of an interesting feature where this gene that has an enzyme that acts in the eye is not actually producing the enzyme in the eye. It's producing the enzyme somewhere else and transporting it into the eye. All right, so I'm going to just jump in to the results is a nice way to go next. And the results are usually depicting graphical results, table results, so numbers or diagrams or microscope images. Starting in this is just a figure of what's known about the structure of the eye cells. And materials and methods, we're going to skip ahead from that one. So pretty heavy stuff. If you are a fruit fly molecular researcher, you might be interested in reconstructing some of these experiments. But for us, certainly we don't need to know how they did everything. Let's get into a little more detail here. So now figure 2 is part of their results. So this is new information, and this is one of the things we want to emphasize. As you're finding papers, you might get a lot of information from the introductions of other papers. But if you want to go to the primary evidence for anything, you need to find the original paper that shows their evidence. And this is really the basis of scientific understanding or scientific knowledge, is having some demonstration of a principle and something that could be reconstructed. So they are highlighting using the staining here that makes this darker black, showing the position of, now we have XDH. So the enzyme xanthine dehydrogenase located in this region right between the retina and the lamina. So dark there. This is a farther out zoom, and then zooming in closer, this is also lamina retina border, and shows that there are these so-called pigment granules that are really housing the xanthine dehydrogenase enzyme, still located at this boundary between the retina and the lamina. Here's more. This is even finer scale within the cells. So these are pigment granules within the cells. And further on, we've got some interactions among mutants. So exploring a little bit more about what is responsible, what other enzymes might be responsible for putting xanthine dehydrogenase into the eye. They have some mutants for other eye colors. So purple, punch, sepia, cinnabar, vermilion, these are eye color mutants. And some of these do or do not have that xanthine dehydrogenase in the eye cells. Here we've got chocolate and prune, full of names. I don't want to go through this in detail right now, but just basically a lot of other mutants and getting evidence of whether those mutants contribute to the position. So a mutant for another gene might not be able to move xanthine dehydrogenase into the eye, and that would be an important discovery to be found, or might not allow xanthine dehydrogenase to do its job. So these are the names for pigment molecules that are made to produce the Drosophila pigments or the pteridines. And some of these, let's see, ones that we study in the lab, here's white. This one's actually white apricot. Is missing a lot of these pigments, so it's mostly white eye color, missing pigments, no big surprise there. Missing, and then lower levels of a couple of these pteridine pigments. Other ones, here's sepia, missing all the way across for this more red pigment, brown, missing across for these red pigments, and don't have any that are totally zero in the pteridine pigments. Okay, so we had the introduction, just a quick look at the discussion. So this is their version of summarizing what was really the most important take home from this result, from this study. Present report describes studies that demonstrate the localization of XDH. So not just in the eye, but in specific parts of the eye, these type 2 pigment granules of the secondary pigment cells of the eye. XDH is synthesized in other cells and then transported into the eye. All right, so kind of an interaction between the brown pathway, red pathway genes. So I guess backing back up to big picture on this paper, as I said early on, we really found mostly what we'd be looking for here is the name of the enzyme, xanthine dehydrogenase. It mentions pigments that it's able to make and pigments that it is not able to make. And for us, that's an important piece. Those are really all the important pieces we need to know for the function of the Rosy gene. So Wild type fruit flies would be able to make those pigments, and mutants that are homozygous recessive for the Rosy allele, the Rosy gene, they would not be able to make those specific pigments, and that explains the rosy eye color. So we could use this information and speculate about what, or hypothesize what a double mutant might be if it was missing the Rosy gene and also one of these other genes in the red pathway or the brown pathway.